I'm so happy to be in this. And also thank you all for coming and being part of this thing. I hope you guys will enjoy what I had to say and learn something from here. Usually I like to have it two-way. So if the internet and everything permits, do interrupt and ask me questions as we go along. I will try to keep posing questions, and I will pause a little bit to see if people are going to answer. And it's possible that due to the lags with the internet, we may be a little slow. So please, forgive me if I don't wait for your answer and I move on to the next part. But we will, at the end, we will make time to discuss any questions that you have. So as Dipti said, I am going to be introducing you, if you haven't already heard, rhythms and how it might impact your interpretations of experiments that you may do, be it behavior, be it physiology, be it cellular, molecular, or genetic mechanisms that underlie aspects of the biology that you are interested in with flies. So let's begin with this movie sort of thing that I would like to play for you, which emphasizes, okay, somebody's in the waiting room. How do I move that? It's moving on, sorry. So I want to show you this movie to emphasize that everything on Earth, the fact that we live on this planet that rotates on a 24-hour cycle of day and night, means that these geophysical factors, I don't know if that music is really loud for you guys. Can you hear some music? I didn't intend to have that. Anyway, the point is that
we can't hear the music, but we can see the video.
Oh, great. So fantastic. Yeah, I'm glad you can't hear the sound. It's a bit loud. So there are geophysical cycles due to the fact that the Earth rotates on its own axis with a 24-hour speed, which means light, obviously, and along with temperature, along with it, humidity, and many, many other factors change. And they may seem trivial because, after all, this is what has been happening to us all the time. We barely even think about it, maybe. But nevertheless, these factors have huge impact on the life and actions of every life form almost that lives on the surface of the Earth for sure, and even in oceans, and even in places where you think directly sunlight might not hit because of the indirect impact of geophysical environmental factors. So what are these things that are changing? Of course, light, temperature, humidity. And because of the fact that these things change, there's also biological factors that change for animals, plants, insects, other microorganisms, the presence of predators, the availability of food, the presence of mates, and so on. You could also think about various biological rhythms that exist. The most obvious ones would be our own activity and rest cycles, our sleep cycles. I'm not sure why I'm not able to change. Okay. The times at which we are able to go out, forage, feed, the times at which animals can go courting for the members of the other sex, mate, give birth, laying eggs, all of these are known to be rhythmic. And by rhythmic, I mean with a frequency that has a speed of 24 hours. Now you could say from here that yes, there are rhythms everywhere. We see rhythms in the geophysical environment, and we see rhythms in biology. So could these biological rhythms simply be our responses to the fact that the Earth rotates on its own axis, right? So the Earth rotates on its axis, all of these things change, and we, biological systems, biological organisms, be it microorganisms or us, simply passively respond to the change. When it's night, I do different things. When it's day, I do different things. Do we really need to invoke a clock, right? Now we have moved from being just looking at rhythmic phenomena to now starting to think about there being a clock mechanism that is behind it, which is not necessary actually. You can think of it as being simply responsive without needing a clock. But in order to, if we are going to think that there are good reasons for us to believe that there are internal clocks, other than the fact that we've read about them, we must have some hints or some hunches that make us think so. Do you want me to stop here and ask you for your hunches for why you think there are clocks? Are these rhythms merely responses to the external cycles? Someone wants to unmute themselves and give me an answer. Otherwise, I will go forward. Or raise your hand or do something so I know there's something going to happen.
Yes, it could simply be a response to biological rhythms if it was consistent and, if the stimuli were more or less consistent. But the fact that there's so much variability associated with it, cloudy days, rainy days, days in which, temperature fluctuations are not a lot, might require some sort of internal mechanism to keep track of it, to average through the noise.
I see. Okay. So I think it is Dinesh who answered. And what I think he's trying to say is that the fact that there is so much variability, it's not as if even though the Earth rotates with an exact 24-hour speed, there are so many other things change on a day-to-day that we can't completely rely on the external cycles. And therefore, it appears that it might be useful to have an internal machine that will help us to know what time to do what. Okay, so I'm going to go with that answer unless somebody else has something else to say. Anything else that gives you a hint? Are there people here who usually wake up before their alarm clock rings? If you have had a good rest and you are able to get your body's requirement of sleep, I think some of us may have experienced the fact that I put my alarm at 6 a.m. let's say every day means I usually already a little awake before months, which makes me think that there's something that's inside me that's already giving me an indication of what time it is going to be. Okay, and you can also imagine that it is helpful for organisms to be able to prepare, let's say for the upcoming dawn or for the upcoming dusk, if they have to do certain activities immediately after dawn or dusk. It's also possibly helpful for your body's, let's say, intestine to be able to prepare the required machinery to deal when food is about to come, or tell it not to make food when it's not supposed to be making food. So it makes sense to think that it might be useful to have a clock, and that the usefulness of it is probably somehow translating into there being some Darwinian fitness associated with it, and therefore it might be helpful to have a clock. And possibly not all the rhythms that we see in biology are merely passive responses to the environment. Okay, so now that leads us to the question of what do clocks do? I want to tell you that there are two things that clocks do that we may not usually think of. We usually have an answer that says clocks tell us what time it is. That is true. Clocks do tell us the local time. But one more implicit job that a clock does is that it gives you an idea of the passage of time. That certain amount of time has passed, that you had to wait for this long to get to this talk, or you have to wait for a certain amount of time that you're estimating before you will be done with this, or when you will be about to get dinner, when you would need to go to class, and so on. So clocks do both of these things. They tell you about what the local time is here in Bangalore, in India, or wherever you are, and also give you an estimate of time passing. That is the job of a clock, regardless of its speed. Okay? But today, our discussion is going to be about clocks with a daily frequency, and we call them circadian for a good reason, and I will come to that in the next slide. Why am I making that
Sheeba?
point here? Yes.
May I ask a question here?
Yes, please.
Yeah. Is this passage of time or the amount of time measured same for the different clocks, or can it differ?
Yeah, it's an
I mean,
integral part of, So the speed of your clock will determine your passage of time measurement as well. So I will
okay.
come to it in maybe in another 2 slides. So these two functions or jobs that clocks do also are reflective of two important parameters of the clock. One, its speed, and second, its phase. So the measurement of the passage of time is intricately connected to its speed. So a faster clock's measurement of passage of time will be different from a slower clock's measurement of the passage of time. Making sense?
Ma'am, I'm so sorry. Could you please repeat the last sentence? Your voice was breaking a little.
Okay. So what I was saying is, what Sujata was asking is, does this measurement of the passage of time depend upon the kind of clock you have? I'm saying yes, in the sense that if you have a faster clock, a clock that runs, so give me a minute. I haven't gotten to that part yet, but I will come to it. Okay, just hold on to this thought for a minute.
Sure.
Okay. Another question? Somebody is saying something? No? Okay, so I said I'm going to be discussing today circadian clocks because I'm sure when I said think about all the rhythms outside, I only talked about daily rhythms, but in fact there are rhythms of various frequencies. But our discussions today will not be about, let's say, heartbeat rhythms or tidal rhythms or lunar rhythms or annual rhythms. Okay, those are there, but at this time we are talking about clocks that are regulating, modulating daily rhythms. And it turns out those are the best studied, but not, I do not mean to say most interesting, because actually there's lots of interesting stuff out there waiting to be discovered about other kinds of rhythms as well. And yeah, huge potential for research. Okay, so we defined our goals, learning about circadian clocks. So I must define what circadian clocks are. By our discussion so far, you have already realized I am talking about entities that lie within organisms. We're talking about biological entities, not physical clocks. They must lie within organisms. You must possess a machine within a cell, within a body, within a tissue that can measure time. Okay, it is endogenous, it is internal, and it has a period of approximately 24 hours. Why approximately 24 hours? Because we are only going to call it a circadian rhythm. We are only sure it's a circadian rhythm if we know that it can tick even in the absence of external cues. Okay, so I will measure the speed of this clock in the information. So there's information received by the organism or by the clock, and find out what is the speed rhythm. It turns out circadian clocks or daily clocks with a daily frequency usually do not have a 24-hour speed, but is usually little longer or shorter than 24, and therefore circadian, approximately a day. So hence circadian clocks. They are innate in the sense that they are governed by I'm going to shut off my video because I think I have poor internet. Are you guys able to hear me continuously?
It's breaking a little.
Breaking in between, ma'am.
Everybody with me, right?
Yes.
Okay. So what I'm saying is that they must be internal. They must have a period approximately 24 hours. They are innate. That means they're coded into your system. And these clocks are ubiquitous. They've been found from bacteria to humans. I said that these can be called truly circadian clocks if these can persist. That means the rhythm that it drives can persist even when you have shut off all information about time from the outside and show a period which is near 24 hours. But even though you have a near 24-hour rhythm, that means it's never 24, but if that rhythm is subjected to time information from the outside, these clocks must be resettable by environmental cues. Otherwise, it's not a very useful clock. Imagine you have a clock and it turns out they are built like that they have a shorter or longer than 24-hour speed, but you live on Earth and earthly environments have an exact 24-hour rhythm. And so if you are unable to reset yourself, then you don't have a very useful clock, right? So internal clocks, circadian clocks, have a near 24-hour speed. They are also synchronizable, resettable by environmental time cues. A third very important feature of circadian clocks is that their period or their speed is temperature compensated. Why am I saying this? Because imagine I just said they are biological entities, which means they are made up of proteins, enzymes, made up of biological molecules, which we know can be affected by temperature. So if the machinery that makes up the clock is modified by temperature such that warm temperatures speed up reactions and cool temperatures slow them down, they would not be very useful clocks. Hence, circadian clocks have evolved ways by which their speed is compensated for even in the face of somewhat large changes in temperature, the ones that are within the physiology of the animal's life. Okay, so this is a very important feature of a circadian clock. Another feature which turns out is there, something to keep in mind, which I will elaborate later, is that these clocks are often displayed at many levels of organization, from the cellular level to the tissue level to an individual level to even at a populational level. Okay, keep this at the back of your mind, the point number 4, it'll come back at some point. But 3 things: that they have a near 24-hour rhythm, even in the absence of external cycles, they have to be synchronizable by environmental cycles, and that their period is temperature compensated are critical features of circadian clocks. With this, I move on to show you an example of what I mean by persistence of this rhythm under constant conditions. And for that, I will take the example, in fact, that is thought to be the first ever demonstration of a persistent rhythm or an endogenous rhythm that was performed by this French astronomer called de Mairan. He has a long French name, but I'm going to call him simply de Mairan, who in 1729 did this experiment where he placed these touch-me-not plants, which we know close their leaves at night, in apparently a basement wine cellar, and found that these plants in the cellar, even in the absence of information about light or temperature, he claimed, from the outside, continued to show a cyclicity, which is a rhythmicity in their opening and closing, even though there was no light coming into the basement or temperature cycling in the basement, which led him to conclude that these plants have a way to know what time it is outside. It remained as such and did not get much further attention other than this report in the Proceedings of the Royal Society by somebody else. In fact, he didn't even report it. But from that point onwards to the early 1900s, there was not too much. But in the 1900s, many different people carried out studies on these rhythms and tried to understand whether or not they were endogenous, and went on to show that indeed these are innate, and also worked out many aspects of these clocks and laid out what I showed you in my previous slides to tell us what is it that can be considered as an endogenously driven, clock-driven rhythm. Okay. So I'm going to go from a plant example to a human example. And here on this schematic is the example of levels of plasma cortisol that is oscillating. You can see a wave in the level. So y-axis is the amount. And on the x-axis is time. And the light yellow shading and the gray shading shows you the time during which this human, was in the presence of light, and M indicates midnight. So you can see that in the middle of the gray shade, and N indicates noon. And so you can see plasma cortisol nicely oscillating. But these people are in the presence of light and dark cycles. So you could think of that as being timed information. But then, okay, and what am I showing here with this red double-headed arrow is that the speed of this rhythm is exactly 24 hours. Peak to peak distance is 24 hours. Sorry, just give me a second. Sorry, excuse me. So this is the speed of the rhythm in the presence of a 24-hour light dark cycle. Okay. Now, what about these humans' plasma cortisol if they did not have information about time? So what happened here is that these humans are now placed in a continuous light situation where food is being supplied to them in an hour and a half intervals throughout the day and night, and they are supposed to remain in a semi-reclined position. And yet you see that these guys will still show you an oscillation in plasma cortisol levels, and the peak to peak distance is now not exactly 24 hours, but has drifted a little. Usually in humans, we find that the rhythm has a longer than 24-hour speed. This tells us that there are certain endogenous machinery inside humans that enables the persistence of plasma cortisol, which is only one out of 100 or other examples of substances in our blood and other body fluids and tissue that oscillates and can continue to oscillate even in the absence of external time cues. So we saw two things, that our bodies have rhythms that can persist in the absence of external time cues, and we saw that when they were in the presence of a light dark cycle, they were able to synchronize to it. So then you might ask, what synchronizes internal clocks? Can they be synchronized by any kind of cyclic time cues? How would I know? So in the real world, there are several things that organisms encounter in a cyclic manner, like temperature, humidity, food, mates, predators, etc. But we don't know that every one of these environmental factors are being perceived by our internal clocks as time givers, true time givers for our internal clocks. In order to find that out, we will have to do certain kinds of experiments to ask whether our internal clocks have been truly entrained to these external cycles. We have to look for what kind of relationship in phase these exhibit with the environment, and how stable is it, how nicely does it lock to our internal clocks, and whether we show any signs of being able to anticipate these external cycles. But for today's discussions, I will not go into the details for now. If anyone wants to know more about this, we will discuss that later. Okay? So I will go on with the understanding that there are internal clocks, they synchronize to external time cues, external cycles of certain kinds that they are sensitive to, and they will reset themselves. Okay. So this kind of understanding had been there, like I said, for several years. In the 1900s, it had begun, and people were investigating many organisms, including Drosophilids, and usually more often Drosophila pseudopoda, not Drosophila melanogaster as yet. Okay, but there were questions about what might be the molecular basis of these clocks, and unlike now, when we have a lot of researchers, converging on many model organisms, it was a time when people were more adventurous about the kind of organisms that they used to study. Okay, so even the study of biological clocks, if you compare it in today's world versus 50 years ago, you will see a huge variety of organisms that people used to study clocks, understand clocks, and even try to ask questions about the molecular basis. And based on our understanding of and comparison of these rhythms to physical clocks, the way we think of how, or the way we know how physical clocks are made, people theorized on how might we be able to generate a self-sustained oscillation. Because remember, what we need for a clock is a way to be able to generate a wave, an oscillation, even in the absence of information about the speed from outside, right? That speed is high fidelity and it must be generated on your own. And the idea was that it would be possible to do that if you have some mechanism to make a feedback loop. Okay, if I had stopped at this and let you come up with this answer simply about it for some time, usually a group of people would come up with the idea that you can get a self-sustained oscillation if you have a way to create these feedback loops, and usually there will be some input of it being a negative feedback loop. Okay, negative feedback loops are most likely to be able to give you something like this. But what kind of negative feedback loops? Again, theoretically, people predicted there might be negative transcriptional feedback, there could be translational feedback, there could be post-translational steps being, being subjected to feedback. There were even ideas that perhaps there is feedback that is happening from the level of membrane electrical activity to internal cellular processes, cellular pathways, signal transduction pathways, and so on. So these remained in the realm of theory, and some level of biochemical analysis was also happening. While this was going on, our hero stepped into the picture. I'm sure you heard in the course of these lectures in the past few days, and he will feature here prominently because he, along with his student Ronald Konopka, were the first to discover the first circadian clock mutation using approaches that I think you guys have already heard about. And this was simple EMS mutations that were carried out on hundreds of cultures of flies, eventually identifying this one locus, which they call the period locus. But before I go to the period, I want to take you away for a minute before I get back to Zina Benzer's experiments, okay? Is it okay for me to go on, or are there questions that you want to stop me at now? Okay, okay. Everybody is quite familiar with this life cycle, but I put this here to show you that flies exhibit many different kinds of rhythms. I will talk about more later, but eclosion rhythm was among the earliest rhythms that circadian biologists had been studying for a long time, even before the genes and molecules came into the picture. There's also a rhythm in activity rest, there's the rhythm in sleep, there's the rhythm in eating, there's a rhythm in when flies will mate, and also when flies will lay eggs. So I hope you guys will all consider this when you are carrying out your experiments. All of these behaviors are rhythmic. All of these behaviors are driven by internal clocks. And therefore, if you are going to be doing experiments where you may make interpretations about levels of any of these, you must make sure that these are being carried out under strict light-dark cycles, and they are sampled at the same time, and all your experimentals and your controls are being treated similarly, not only spatially, but also temporally. Okay. Let me explain to you a little bit about how we record activity, after which I will take you to the eclosion rhythm, which is actually where our story will continue. Just a slight deviation. So what we do, and several people in India also carry out these studies, where you can put these flies into these little glass tubes. So an individual fly remains in this glass tube with tissue paper plug on the other end. And these can be placed into what are known as Drosophila activity monitors. These were the old kind, but now Trinetics, which makes these monitors, have come up with one more type of apparatus. I will show you that a little later. The principle is that, can you see my pointer? No.
Yes, ma'am.
Yeah.
Okay, okay, fantastic. So what happens is that these tubes are laid out on these monitors, and in this gray region which you see, no, there's actually a pair of infrared emitters and sensors, such that now if this fly is sitting here in this tube, there is an emitter and sensor giving out a beam of IR. So whenever the fly moves, this beam of IR is getting cut, and whenever it's getting cut, it is being taken as a count, a read account that is fed into a computer. And so you can do this for as long as the fly is alive. And you can do this inside incubators or wherever you want to do it, and modify the kind of environment that the fly is facing. So you can either place it in complete darkness and ask the question whether this rhythm is endogenous, or you can give it light dark cycles of various kinds and ask, does the light dark cycle synchronize the activity rest rhythm of my fly? Or you can take it out in the natural world and ask, compared to summer and winter, how does the rhythm look? So these are the different kinds of experiments that people have been doing using these monitors. And as I said, there's another version, a standing version also of the same monitor available. Here also the same, except that you have now two pairs of infrared emitters and sensors. And I hope you can see this fly sitting here. So whenever it moves, it cuts this beam, okay? And what you get at the end of it are these records of activity, which looks like this, which we call an activity rest graph or an actogram. So if you encounter papers that have anything to do with rhythms, you will see something like this, which is why I will take you a minute to explain what this means. This is basic for okay? One individual fly on the x-axis 0 to 24 hours is plotted. On
I think your voice is breaking.
day 1, day 2, day 3, day 4, and so on is plotted. And you see a black vertical. Okay, so whenever you see a vertical beam, vertical bar, it means that at that time, that means around 8 o'clock, the fly was active and then became less and less active and was at rest. And later on in the again started activity at rest. And they remained at rest. The next day they started their activity at around the same time. And they did that day after day after day after day after day. On set, now I did not tell you what kind of light regimen were these flies in. So it turns out this, yeah.
Your
Yes,
voice is
Deepti?
breaking.
Oh no.
Voice is breaking.
Yeah. Can you switch off your video?
Let me see if I shut off my video. I will do that.
Yeah, yeah.
Okay. So I was saying that this is a record of the activity rest pattern. At particular example, the fly has been experiencing 12 hours of light during this time and 12 hours of darkness. The dark shade indicates night and the light shade indicates that they were in the presence of light. So if I drew a line that is going from one day to the next of set, you will get an exactly 24-hour rhythm. I do such now light, and you will see once to the fly now starting from day 11. Watch what happens. Across days right. To make this easier on the eye, what I am going to this
Oh, I think we've lost Sheeba. Let's wait till she joins back. Sorry about that.
Yeah, sure.
Am I back?
Yes, you are, Sheeba.
Can you stop my video with Deepti if you want to? No. Okay, great. Thank you. So this is an example of an actogram. And you can see that when the fly was in the presence of a light-dark cycle, they show exact 24-hour rhythm. But once they were released into continuous darkness, now the rhythm has a greater than 24-hour speed. And this is a reflection of internal clock because there is no information about time from outside. From the activity rest, we go to the rhythm because that is the rhythm that helped Konopska and Benzer to be able to identify the period mutant. But it needs to be admitted. On left, you are seeing a plot of closing flies in a culture. Flies are axis is day 1, day 2, day 3, 4. On the y-axis is how many flies have emerged per hour. Those of us who have experienced collecting flies in the lab, if I want to do virgin collections, usually I will try and make sure to come in just around when lights come on. I'm placing my flies in a duck cycle because I know that flies will maximally emerge just after lights on. But what you're seeing here is that when these scientists place these flies in continuous darkness, also flies showed a rhythm in emergence such that flies maximally emerged at one time of day and continue to do so on other days as well. This, if you estimate the speed of this rhythm, you will find that it has a speed of approximately 24 hours. What Benzer and Konopka was looking for is whether subjected culture to EMS mutagenesis, do I see any deviations from this normal eclosion rhythm? And what they saw was indeed that they find 3 deviants. Here is a deviant which clearly is not rhythmic at all, which they termed arrhythmic. And here is a deviant who finishes 4 cycles of eclosion by the end of 3 days. The peak distance is much less than 24 hours. And so the rhythm has a faster speed or a shorter speed than your controls. On the other hand, here is the third mutant or third deviant where the 3 days have passed and yet 3 cycles have not been completed. Distance is much longer than 24 hours. From these studies, from these cultures, Sheeba Benzer and Konopka also took individual flies and placed them into activity monitors and found that while controls showed this rhythm of near 24 hours, the arrhythmic individuals showed arrhythmic cultures had arrhythmic individuals with arrhythmic activity. The short period mutants had a short in their activity rest rhythm, while the long period mutants had a long period in their activity rest rhythm. This show, and I forgot to mention that they also did the necessary kinds of complementation experiments along with deficiency lines and did staining experiments and so on, and were able to show that all the 3 mutations are lying on one locus, which they termed the period locus, and is considered as the first clock gene to be identified. Okay, so Sheeba Benzer and Konopka in 1972, published this paper that announced the discovery of this locus that is critical for generation of the rhythm and for determining the speed of the rhythm. So you can see that mutations at this locus has made either the flies go completely arrhythmic, bazooka, right, gone. So a period, no period, also able to determine the speed of the period, short versus long. So they knew that they have hit upon a locus on the X chromosome of the fly, which is able to hit a part of the clock which is needed for it to become a clock itself and also affect its speed. But it remained as such until these guys came along, Jeff Hall, Michael Rosbash, and Mike Young. And some of you may be aware that in 2017, these three were awarded the Nobel Prize in Physiology and Medicine for their discoveries of this underlying cellular molecular basis of circadian clocks. So the work that these guys carried out under intense competition and rivalry resulted in our understanding how these molecular clocks are built. And I'm going to sort of say to you in a nutshell, the work that was done over a period of maybe 20 years. Okay, so bear with me that I compress all of that information and put it in the form of a somewhat simple model. Okay, I also will quickly tell you how did they go about this. So I am sure you guys might also be thinking that if this is a gene which is so important to be a clock, and after all, clocks are made rhythmic event, that the clock gene itself might be rhythmic. Isn't that a reasonable assumption to make? So after they did these mapping and sequencing and rescue experiments, the things that they next tried to do was to generate antibodies against the period protein. And lo and behold, they were able to find that, yes, this period protein actually localizes to specific cells in the brain. And moreover, in Western blots, they showed protein levels nicely oscillates, and that oscillation has a daily rhythm, and that will continue to happen even in darkness. So that was already a big satisfaction that you have gone from just knowing the locus in the 1970s to early 80s, showing that there is a oscillation in the period protein. Then they also saw that the mRNA levels also oscillate. That was possible only because of the ingenuity of one of the people from Michael Rosbash's lab, who was actually an expert in RNA biology at that time. And his postdoctoral fellow, Paul Hardin, discovered that the mRNA level of period in the head of the fly oscillates. And he was also able to show that if you can make mutations in the period gene in the background, so if you have two kinds of alleles, you can show that certain can override the wild type and influence the mRNA oscillation. Another interesting factor was that while the period protein oscillates and the mRNA oscillates, they saw that there was a beautiful 6-hour lag in the oscillation peak of the protein compared to the period mRNA. So this is not all that intuitive, because it is very rare to find that a protein lags so much from its mRNA. And that they also saw was that the period protein, so this will be able to do immunohistory on slices of fly's brain, able to the cells cleanly and where the protein is present in the cells, and they were able to see that if you sample these slices across time of day, that this protein will shuttle between being in the nucleus and the cytoplasm. So I said, first of all, the levels itself oscillates. Secondly, it is present in the cytoplasm for a certain amount of time first, then it goes into the nucleus. After a while, it is completely absent. Now this gave people the idea that since it is likely for a clock protein to behave like a clock protein, to be involved in some kind of negative feedback loop, does this mean that the per protein has some way of being able to inhibit its own production? Is that making sense? Okay. The fact that it's able to enter its nucleus and it does so only at certain time to come up with this. Such studies were
Sheeba, your voice is breaking. Hello,
I
Sheeba.
have, hi, I know I got shut off again.
Yeah.
I'm going to I switched to another device, in the sense a hotspot, and I hope this will be better.
Sure. Yeah. Thanks.
Okay. Did you guys get what I was saying until this point, that a bunch of different genes were discovered using similar approaches? Check. Check. Deepti, can you hear me?
Yeah.
Okay. Can I continue from here?
I can hear you.
Yeah.
Sure.
Can I continue? Yeah, from here. Okay. So using a similar approach, many different genes were discovered, and not just that they were discovered, a bunch of papers again from these three labs showed the interactions between these genes and their proteins. And I'm going to summarize a model of how these proteins and genes might interact to form a self-sustained oscillation, which involves negative feedback loop. Okay, so on the left of this cartoon is supposed to be a nucleus and the cytoplasm of a cell where there is a clock residing. And imagine what I'm drawing here to be a cartoon of the regulatory region of the PER gene. So this white box is supposed to be like the business end, this coding part of the gene, and on its left are the regulatory sequences for PER. So PER gene will get transcribed and PER mRNA will be produced only if a heterodimer, that is composed of two proteins, clock and cycle, come and bind. So upon their binding, PER mRNA gets made. PER mRNA in the cytoplasm will make PER protein. Similarly, on the timeless gene, imagine this cartoon to depict again now the timeless gene, regulatory sequences of the timeless gene, EBOX sequences. Again, clock and cycle must bind to that region such that now timeless mRNA must be made. What I'm saying is that both PER gene and timeless gene are similarly regulated by this heterodimer or transcriptional activator complex of heterodimer of clock and cycle proteins. Once transcriptionally activated, PERTIM is made, PERTIM mRNA begins to build up, but it takes a while before PERTIM protein builds up, because PER and TIM proteins by themselves are highly unstable and will be stable only if sufficient number of them manage to form this heterodimer. Once the heterodimer is formed, further they must become phosphorylated for additional stabilization. Once that happens, they can enter into the nucleus. Once they have entered into the nucleus, also they are subjected to further phosphorylation, attacks by other kinases. If they manage to escape all of that, then the PER-TIM heterodimer is able to go and upstage clock cycle and prevent clock and cycle from transcribing their own mRNA. Okay, in this manner, PER protein and TIM protein are negatively inhibiting their own transcription. And this, one can imagine, can go on under the assumption that there are no major shortages of any kind. This whole thing, you can imagine, could be completed at a time scale of 5 hours 5 minutes or longer. However, it turns out it happens within a duration of about 24 hours. Why is that? That's because there are several breaks and checks along the way. I have not shown all of them, but there are many phosphatases and kinases in the mix that can prevent parenchyma protein from reaching sufficient levels and being able to go in and inhibit themselves. And the only way, or the stable system has evolved that allows them to complete this loop in 24 hours. And this is referred to by chronobiologists or circadian biologists as a transcription translation feedback loop mechanism, or a TTFL. Now, I am describing this as if it is happening in the absence of any time information from the outside. Now let us bring light into the mix, okay? Because after all, this whole process is happening in the presence of a light-dark cycle. So it turns out this process of parenchyma mRNA making parenchyma protein and parenchyma protein building up does not happen because timeless protein gets degraded by light, not directly, but because of the presence of a photoreceptor called cryptochrome. If cryptochrome degrades timeless and puts it away into the proteasomal machinery to be degraded, per protein is by itself. Per protein by itself cannot remain stable, gets degraded, and so this whole process has to wait for nightfall. Only in the absence of light can the timeless protein and per protein become a stable heterodimer, get phosphorylated, enter into the nucleus and inhibit its own transcription. In this manner, even though there is an approximately 24-hour rhythm of the internal clock, the light cycle ensures that there is an exactly 24-hour rhythm. Okay. This, like I said, is a very highly simplified situation. And in reality, there is not one loop, but many different loops, negative and positive, interlocked loops. And the beauty of this system is that so please focus on the top. Here is period and timeless protein, nuclear translocation, period and timeless protein inhibiting clock and cycle normally making parenchyma mRNA, parenchyma protein, heterodimer, etc. This is the loop I described, but I said there are also other loops that are interlocked because of common players. The beauty of the revelation of this machinery is that similar machinery has been identified in mice, in humans, in plants, in fungi, in zebrafish, and in cyanobacteria. And that is the reason why these three scientists have gotten the recognition that they did. The fact that they uncovered, with the help of fruit flies, a molecular machinery that seems to have independently evolved in many organisms and has the basic similarity in structure. Okay. So I'm putting this in a nutshell, or in a summarized form, that this TTFL that I was talking about, the transcriptional translational feedback loop, is considered as a core clock machinery for the most part, okay? Because there is transcription translational based feedback. In addition to that, there have been discoveries of feedback loops in the cytosol, which involve many such molecules like calcium, CAMP, NAD, NADH, AMPK, and even glucose. In addition to all of these, there have been several other metabolic flux oscillations that have been identified that I haven't put here. And the way we understand it now is that in cells, possibly all of these mechanisms exist and possibly interact with each other. Okay, I'm going to go from here to, okay, shall I stop here and move on, or do you have questions?
So there
You
is
have
one question which is from, I don't know who, from somebody.
Okay.
Where does the melatonin come in all this?
Oh, give me just a minute. I'm moving right there. So all of this was at this point simply inside a cell, let's move on to ask where then. Good question. Where do these clocks lie? Okay. So melatonin, you said. So here is my human slide. Okay. Just to put this in perspective. So in humans, it is thought that there is a central clock that lies at the base of the hypothalamus in the structure called the suprachiasmatic nucleus, written over here. It sort of lies over here, and the suprachiasmatic nucleus has a self-sustained oscillator, okay, similar to what I described to you some time ago, which means it can generate molecular oscillations on its own, but that is only its internal clock. It is synchronized by the external world, mostly by light in humans, because light that comes hit our eyes through certain special cells, including visual photoreceptors and non-visual photoreceptors, through certain optic nerve pathways, reaches the suprachiasmatic nucleus. Once the suprachiasmatic nucleus gets this information, the internal clock can be reset by light. Then information from the suprachiasmatic nucleus reaches many other parts of the brain. That slide, that picture I do not have here, but many other parts. But in this diagram, you can see that it certainly shows you how there is a connection to various endocrine organs, including the pineal gland, which then makes melatonin, which then reaches to various parts of the body. In addition, the SCN communicates to other parts in the body where also there are clocks. So while we used to think of there being a clock only in the brain, we know now for many years that that's not entirely true, that the clock machinery in fact exists in several tissues. And some of these tissues can, under certain circumstances, simply ignore information that is coming from the SCN. Usually what happens is our body does not have one clock, but you can think of it like a clock shop, such that there are clocks all over in various tissues. But the job of the suprachiasmatic nucleus we think of as the conductor of an orchestra, where if there are many instruments playing different rhythms, this SCN helps to synchronize these rhythms and keep them appropriately phased with each other. Okay, so various tissues throughout the body have clocks. There are certain somewhat more dominant clocks that are present in the SCN. For certain functions, they can override. So you see all these lines emanating from the brain to various tissues. There are also, you can think of these lines also going back to the brain. Okay, so there is also feedback from these peripheral structures to these central clocks. So we think of these as being peripheral and central oscillators, and them all being able to communicate nicely with each other to set our rhythms in sync. Now with this, I'll bring you to the fly brain and show you with this cartoon, some cells that are thought to be core pacemaker cells in the fly. Out of them, these cells called the small LNV had been for many decades, not many decades, okay, couple decades, thought to be crucial pacemakers. But now that also is being challenged, and we now think of it more like a network of oscillators that control rhythms, okay? The parallel I want you to look at is that here in the vertebrate brain, here in a mouse brain, also the suprachiasmatic nucleus that lies over here, shown here in green, also communicates through two other parts of the brain and also receives inputs from other parts of the brain. And thus, we have some parallels in the network organization of clocks. Okay, I've actually gone into 1 hour, but Deepti, do I have time to actually, because I haven't even gotten into this, will I be able to extend for maybe another 15-20 minutes?
Yes, yes.
Okay.
Yes, yes.
Okay.
Absolutely, absolutely. Yeah.
Okay, should I like stop for a few minutes and maybe stop and take questions?
Yes, there is one question. Sneha is asking that you have talked about involvement of light in the molecular pathway.
Yeah.
How then is the rhythm maintained in absence of stimuli like light?
Okay, so I was hoping to be able to convince you that even without a stimulus, okay, a negative feedback loop can give you a rhythm, right? You are talking about the persistence of the rhythm, if I understood right, no? Did I get the question right? So what I'm saying is, it is one of the fundamental features of a clock is that it should be a self-sustained rhythm, and a negative feedback loop can give rise to one. Okay, it just happens that loop gives rise to a near 24-hour rhythm, and on a daily basis, if it is little longer, it is reset backwards. If it is little shorter, if it's a little shorter, it is reset backwards. If it is a little longer, it is reset forwards, such that if imagine I have a watch, okay, and my watch is always running fast, but I live on Earth and I have to set myself to a 24-hour rhythm. So every day when I find out that I'm off by half an hour, I simply reset it backwards. On the other hand, if my clock is running slow, every day at a certain time, when I know, let's say Akashwani tells me that it is 8 a.m. or Vivid Bharati tells me it's 8 a.m. or something else tells me it's 8 a.m., I use a reliable information from the outside that I can be sure it is 8 a.m. and then I reset my clock appropriately. Okay. So the light agent is a resetting agent. Now, if your question is about the final output, which is the behavioral rhythm of locomotion or activity or sleep, I haven't gotten to that yet, but these are the rhythm generating agents which will eventually have to give rise to your output. Am I making sense?
Yeah, ma'am. Thank you.
Okay.
Yeah, answer. Yeah.
Oh, thank you.
Follow-up question. So the melatonin, which you said, so is that equivalent to the cryptochrome? Is it the one which is actually resetting the clock? So is that spread? I didn't quite get. So this melatonin, that seems to be the only thing which is actually talking to the outside world. The light seems to.
Yeah, that is misleading because of the cartoon I'm presenting to you. Melatonin simply happens to be one of the well-known agents. Look, everybody, I mean, you guys came up with melatonin without me having to say it, right? It's something you study in school. It is a neuroendocrine substance that has very broad effects, including affecting your sleep. But it is not the only one. By no means not the only one. That is why this cartoon shows you so many arrows coming out from there. Melatonin is only one such. There are both nervous as well as endocrine outputs from the brain.
I see. So, but in the older sort of the Drosophila stuff you showed one, I think you called it the cryptochrome, which actually breaks the cycle and makes it synchronous with outside, with the light, right?
Yeah. So imagine it like this, okay? Imagine three parts to this whole business. There is this central clock, okay? The central clock is an endogenous time generating machine, okay? And that time generating machine has a near 24-hour speed. It needs to be reset by information from the outside. So that is input to the clock. Imagine that here. So information about time from the outside needs to reach the clock. So in this input pathway is where cryptochrome comes for us, okay? Now, not us, for flies. I must make this distinction that cryptochrome in fruit flies is merely a photoreceptor. Okay, it is the way with which light information reaches the clock, and I will tell you in a couple more slides the same point. However, cryptochrome in humans, in vertebrates, is actually a part of the internal clock. I know it's confusing. Too bad. This is how they named it. But the cryptochrome protein in vertebrate clocks is a part of the core clock machinery. Whereas cryptochrome, originally discovered in flies, originally discovered as a relative of a plant protein, it's a phytochrome relative, is a light receptor in the insect clock, at least in Drosophila melanogaster. Okay, so it is in the input. and cryptochrome lies inside the core clock in vertebrates. It is a part of the core clock. Now, melatonin is actually an output. It is on the third part. It is the part that will eventually give rise to our rhythms in sleeping and waking. Do you see this three-part nature?
Yes, I see that. The question I was asking was more the interact. So as you said, if you just think of the vertebrate system, you're saying that the cryptochrome is related to the internal part of the clock.
Yeah,
right? And that makes sense, because what you've essentially told us right now is that there is a clock on in basically every cell, right?
Yes.
Now, ideally, not all cells will get information from the outside world. There'll be sense organs which are actually sensing this.
Yeah,
so
depends
you would
on the
want
organism.
Most organisms, I guess, apart from bacteria, apart from single cell organisms, I guess most multi-cell organisms, not all of them get information from the outside world. Right? So there has to be some transmission which happens. Now, so that's the question I was asking, which is that you would ideally want one location where that reset happens, and all your light, temperature inputs should come somehow and hit that, so that synchronization happens throughout the whole body. And the question I was asking is, how does that happen?
It doesn't happen. You wish it were so neat, but it doesn't happen like that. So what I'm saying is, there is no such one location, as you can already see from here, and which is also, I mean, even within a cell and also here, there is no such thing as one location. So there are some locations which will be more sensitive to light, and some which are insensitive to light, some which are sensitive to food, some that are not so sensitive to food of the clocks.
I see.
Yeah, so it is not as simple as that.
Just taking that logic forward, if what you're saying, if this is what is the case, then ideally when you're looking, let's say, at the body, you would see some systems which are in sync, some which are not, some which are delayed, right?
Yeah,
that's what you
indeed.
see.
It is.
Oh,
yeah.
That's cool.
So the phasing of everything is not the same, indeed. I don't have a picture for that, but yeah, you will be easily able to find that if you look at any broad reviews. Yeah, that is true. That is true.
Thanks. That makes sense.
Yeah. Okay. So, okay, if we have no other questions on those matters, we will move to the fly now. So a similar cartoon is what's shown here in flies, showing you the various parts in the fly head where it is known that such clock machinery exists. So you can see it very nicely in the eye. All the photoreceptors have the clock machinery. There are cells in the antennal segments. There are cells in the ocelli and in regions that are important for food intake. And here you're seeing the section of a brain, and you see there are about 150 odd cells in a fly's brain that contain, now I'm not talking about the eyes, outside of the eyes, that contain this machinery that I described to you a few slides ago. And these are considered to be circadian pacemakers. Outside of the fly's brain, here in this cartoon, you see that such clocks also exist in the fat body that is distributed in these creamish structures, in Malpighian tubules, and also in the ovary and so on and so forth. But what this schematic is showing you is that such organization exists in these different cell types. They are more or less similar. They may have some slight deviations as to what the partners are in those loops and so on, somewhat small deviations. They may also be differences in how sensitive they are to certain, like I told you, inputs. But the details of that I will not go into at this time. The point of this slide is to show you that clocks exist even in fruit flies in different tissues, and they are differentially susceptible to different kinds of cyclic inputs. Okay. I will talk a little bit more about the activity rest rhythm and the circuits that govern the activity rest rhythm, because a little more is known about it. And I also have a little bit more involvement in those studies. So I'll spend a few minutes to tell you a little bit about those circuits. Okay. Here is the fly brain neuronal circuit. In the previous slide, I was simply showing you the cell bodies. And in this beautiful picture by Charlotte Helfrich Foster, who has done studies on this system for many decades now, shows you that these cells are all connected with each other via axonal and dendritic processes. Okay. And this cartoon again by her shows you that different cells in this network express different neuropeptides and neurotransmitters. Among them, the neuropeptide pigment dispersing factor PDF is a 18-mer, which is the best studied neuromodulator of this system. Okay. Other than that, there is IPNamide, there's glutamate, there is also SNPF, NPF, there's also ITP, ion transport peptide, that was more recently discovered, glycine, DH31, and acetylcholine, that are all known to be in the mix in some form or the other and communicate with each other. And these cells communicate with each other. This cartoon shows you that in the fruit fly, light is perceived in multiple ways by this circuit. What are these multiple ways? Light can come through the traditional photoreceptors. The R1, R6 are lined here. 7 to 8 are lined below. They give light information to these clock neurons. There's also the HBILET that also gives light information to the clock. There's light information coming from the oscillate to the clock as well. In addition to all of this, there is also light that is directly received by these cells. Why is that? How is that? How do we know that? You can have mutants which have lost all of these photoreceptors, 1, 2, and 3, and still you can get flies to synchronize to light dark cycles. That's because this molecule, this protein cryptochrome, which is present in these small and large ventrolateral neurons and a few other neurons within the central brain of the fly, can directly receive light, because it looks like the fly's exoskeleton is porous enough for light to reach these cells and does not rely upon, does not need information that comes via these visual photoreceptors. So in multiple ways, light information reaches this circadian circuit inside the fly's brain. Okay. Now I've pared it down again and you have only the cells. And you might wonder that all I have said so far is that there is a molecular clock in terms of these transcription translational feedback loops. How is that then translated, or how is that information then communicated to the rest? So one of the things that would be nice to know is if there is anything about the electrical properties of these cells that also has a rhythm flavor to it. Okay, is there a day-nightness or a time of dayness to the firing properties of these neurons? Okay, so in order to do that, what has been done is to actually put an electrode in and measure the firing frequencies of these neurons. So in addition to simply measuring them at one point in time, what we did was to actually record the electrical activity of those neurons at different time points in the life of this fly, while it had been for several days in continuous darkness, which means we know now, based on the activity rest pattern of that individual fly, that I have sampled this particular fly at a point when it was about to offset its activity. Okay, and you can do that at various other time points, and then you can plot what about its resting membrane potential, firing frequency, what is the possibility of it being a burst versus a tonic firing cell, and so on. And you find that there seems to be a day-nightness to these membrane properties. In addition, the other thing we also saw is that they are sensitive to light, and that sensitivity to light is mediated by cryptochrome. We'll go past that quickly. And I want to show you this study, which was performed by a group in Yale, which showed that this rhythmicity in the firing properties of these neurons is also dependent upon the internal molecular clock. Because of what these guys did was to carry out such experiments in a wild type versus a per null mutant. And what you see is that this rhythm, rhythm in the RNP that was seen in the wild type flies flattened out if you did these experiments in a per null mutant, which suggests that the pattern in firing frequencies that you saw in these circadian pacemakers is reliant on your internal cell autonomous clock machinery being nicely rhythmic, or at least that it is dependent upon the period protein. Okay. So what I did so far, I began by showing you the discovery of the gene. Then I told you about how many such genes and proteins were discovered, how they were all modeled into how they could make a negative feedback loop. And then I showed you where it is present. And then I said that those neurons do exhibit certain features of their firing and about their membrane. That is suggestive of those properties also being governed by internal clocks. Okay. Now I'm stepping back out and looking at this circuit and asking what are the other kinds of rhythms that this circuit controls. I have 528. Okay. Okay. I understand I'm sort of crushing everything into a nutshell. Okay. But anybody who's interested, happy to talk to you about this later. So these gray cells that you can see, in this cartoon are all those 115 cells I was talking to you about until now. But all these other colored structures are new players. Why are they new players? These are the via media through which other rhythms, including of course activity, are controlled or not controlled, are driven by these circadian pacemakers. So let us start with the rhythm in activity, because that is something that I have already talked to you about. So the rhythm in activity, I said, is indeed generated by these gray cells, the circadian pacemakers, among which, if you look at this part of my pointer showing you, these S, L, and B are considered to be really important. And you see by the arrows that the S, L, and B is communicating in this region. You see all these gray arrows going forward, talking to other clock cells. But in addition to these other clock cells, they are also talking to this kind of cell called the leucokinin cells. And leucokinin cells via other cell intermediates also talk or communicate to this region called the bulb region by what is known as a TU-BU interface, okay, in intermediate. And they in turn communicate with certain parts of the central complex in the brain, ultimately affecting activity. Also, these clock neurons talk to through other intermediates to other structures again in this ellipsoid body and control sleep. Another thing that happens is these cells speak to what are known as the PTTH expressing cells, prothoracicotropic hormone producing cells, which in turn communicate to a structure called the prothoracic gland or the PG gland, which eventually controls the rhythm in emergence, adult emergence or eclosion. In addition, these clock cells communicate via ILP, which you heard about previously from Jishi, DH44, CYF-A, etc. to control rhythms in metabolism, in feeding, and also in activity. In addition to all of this, it's known that clock cells, through some means or the other, speak with also mushroom body neurons. So one more cartoon to sort of show you something similar. So here, again in gray are the 150 circadian neurons. They communicate to various other things. One of them is, like I said, via PTTH, prothoracic gland controls eclosion. Second, via the fat body, controls feeding. Third, controls locomotor activity through various intermediates. Controls courtship, I did not tell you about. There are PDF expressing cells in the abdominal ganglia, which via the enocytes also determines the rhythm in courtship. There's also a rhythm in temperature sensitivity that adults have, that is controlled by cells that are sitting at the base of the antennae called the AC neurons, and so on. Okay, this is a bit abrupt, so I'm going to
Hello,
jump.
Sheeba.
Yeah.
Could you elaborate upon, on the crosstalk between these cells and mushroom body?
Okay, actually, the reason why I don't want to go there is that those mushroom body studies, no, it happened a couple years ago, and after that, because we are not, we don't really, we don't have a good handle of the connection between circadian neurons and the mushroom body. So earlier studies implicated that circadian neurons via the mushroom body control sleep, okay, that there is a sleep effect. But now we think that actually possibly the sort of drivers we used were mushroom body and other cells. That is why now we remain within the central complex and the ellipsoid fan shape, ellipsoid body and fan shape body cells rather than go to mushroom body itself. It's a bit murky. That's why I don't want to go there.
Oh, okay, thank you.
Yeah. Okay. I don't, okay, I just skip this thing, okay, it's, I think, unnecessary. I will leave you with this last idea, which I think is very interesting, quite fascinating. And I think all of you guys, I know you've come into this workshop on Drosophila and have mostly probably been hearing and thinking about Drosophila melanogaster, but I'm a firm believer that it's, we should start looking outside of melanogaster, and then we learn about, we understand melanogaster better, because the fact of the matter is that there are all of these Drosophilid species out there with so many interesting quirks and variations that will teach us about biology a lot more if we start looking at them now that we have this, this treasure called Drosophila melanogaster. So this study, again by Charlotte Helfrich Foster and her group, have been looking at other species of Drosophilids, those that have been in the tropic, subtropic regions, those that have now invaded temperate regions, and those that have gone on to become subarctic species. And here in this very nice study, what she does, several other studies, but this particular one I picked, where you can see a comparison of, this is of course a schematic, of the activity of two very different species. Zaprionus indianus is a species that is found mostly in the tropics, and under a 12-12 LD cycle, its activity rhythm looks like this, where there's a nice peak around lights on and another peak around lights off. Drosophila montana, on the other hand, is a very temperate species, remains a very temperate species, and they are mostly active only during the summers in those regions where the photoperiods are long and there are cooler temperatures, and they show an activity waveform that is quite deviant and very different from the waveform that most likely the ancestral species exhibits. So the question could be, what has happened to its underlying clock, what has happened to its underlying neuronal circuitry, and this very quickly shows you how the neuronal circuitry, this is a cartoon of the circuitry in melanogaster showing both cry and PDF expressing cells. This picture here, I'm only showing you PDF expression, and already you can see how there are, major differences in the neuronal circuitry of these three species, which she explains also. She's able to explain using this circuitry why these three species show the kind of behaviors they do. The original paper will tell you more about what she finds. Okay, so I will go now to my take-home messages. Number one, I hope I've shown you or reminded you that these are endogenous self-sustained systems with temperature compensating ability that can stably entrain to external environmental cycles, that they are composed often of transcription translational feedback loops, and that in many metazoans, including flies, that they often reside in special cells, and these cells have rhythmic electrical properties. I hope I've also shown you that in addition to there being these central clocks, that clocks are often present in many other peripheral tissues and that they must communicate with each other. That also, hopefully I have given you this idea that rhythmic processes and phenomena underlie almost every life activity that flies and other organisms exhibit, and that looking at the evolutionary trajectories of some of these phenomena are likely to be very interesting future endeavors. So I thank you for your attention, and I'm happy to answer any questions you may have.
Thank you, Sheeba. I'll just take the questions from the chat first, and then we'll open it. Yeah. So the question from Sujata Deshpande is: Are clocks based on neurons mostly? What about organisms which evolved very early and have no neurons, like before Cnidarians? And then how do single cell organisms choose clock?
I'm sorry?
Do single-celled organisms choose clocks?
So yes, indeed, clocks are not restricted to neurons by no means. Single-celled organisms have these, have the machinery. Plants have them, and so the question is, if there are not neurons and there are still multiple multicellular organisms, how do they communicate, right?
Yeah,
they will produce substances that will move through to other cell types which do not have clocks. But if you look at plants, for example, clocks are in every cell. So the synchrony is probably achieved by like gap junction types of situations rather than relying on neuronal communication. Okay, I didn't tell you that. This also, that even in fruit flies, in the fly circuit in our own lab, we do discover that there seem to be electrical synapses in addition to chemical synapses. Now, single-celled organisms, within the cell, this core machinery exists. So they don't really need to communicate to another cell. So it's sort of self-contained. So within a cell, I think it's okay, right? But you're saying non-neuron containing organisms, do they have clocks? Like I said, even in the fly, you can have fat bodies having clocks. You do not need a nerve cell alone to be having a clock. Yeah, it's not necessary.
Akanksha is asking, what is the basis of jet lag, and why some people experience severe jet lag?
Yeah, jet lag, right?
Lag.
Yeah. So the basis of jet lag is
and
that
why and
yeah, I got the question. Why some people experience more severe effects of the jet lag, I think is what she's saying. One is that your internal clock has its own period, but I said is resynchronizable, resettable. So if you shift your clock to a different time zone, it will take its time to synchronize because your clock is still stuck in India time zone, whereas your jet has taken you to America in a time that your clock has not been able to deal with. It is still stuck at an older phase. If instead you had traveled to America on a ship, which took you several days to cross several time zones, you would not have experienced it because your clock got the time to reset itself gradually. Here it is now being forced to reset across several time zones. So it's like moving your clock by several minutes too quickly. And therefore you experience jet lag because your internal systems are now needing to adjust to this new phase, forward or backward. And the delay in being able to do so is giving rise to the discomfort because you're forcing yourself to be awake and do the things in the United States, for example, whereas your intestine is still not realized that now's the time to make the juices or not make it, which is why often the first thing that people who experience severe jet lag feel is their gut, not behaving. It's one of the most obvious things, but many other things are also out of sync. Why do some people take more time or experience more severe jet lag? Before I say some people, I will tell you that it is also which direction. So our human clocks usually have a speed longer than 24 hours, okay? On average, humans have a greater than 24-hour rhythm. We find it easier to deal with eastward travel rather than westward travels because our clocks are more easily able to shift in that direction. Therefore, if you are going to a conference from the UK, from Japan, or from the United States, the people who came to the United Kingdom from United States will find it easier compared to those from Japan, simply because our human clocks are set in that manner. The other thing is, that is one. Second, let's say we are all of the same, having all of the same, direction of travel, but still have differences in how we reset. It's because of the flexibility of our clocks. Some clocks are simply more flexible and more easily adjusting, just the way they are built.
I hope that answered the question. Falak is asking, which I did not understand. My question is, after getting the light and dark data by DAM, how we make the graph?
Oh yeah. So luckily, the guys who make these DAM monitors also have a easy way to give you the data out in the form of a notepad file. Okay, and now many people have developed free versions of applications, including my lab have developed 2 free applications with which you can make these graphs. There are paid things as well in Clock Lab, which uses MATLAB and so on, but there are several free versions. You can get in touch with me if you need to do that. And the DAM guys, that is Trikinetics, on their website have also featured many of these, which include our software, our app.
So Falak, you can get in touch with Sheeba.
Yeah, sure.
For this. Ash Tabi is asking when to flip the flies such that they are most active while performing experiments.
When to flip the flies such that they are most active. Okay. Actually, if I understand, flip the flies means when you get them to lay. And
I guess when to when to perform the experiment.
When to perform. Okay. When to perform. Now, this is a very tricky thing because when to perform is not something I can really tell you, because if you're trying to, that's tricky because you may, ideally, you have to do the experiment at multiple times of the day, okay? If it is an activity that the animal does while it is awake, if you don't know, you need to find out. But if you have neither the time nor the inclination, at least you should say, okay, I know flies are maximally active at dawn and at dusk. So if I'm trying to do an experiment about that is testing the alertness of a fly, I should not be doing it at night. I should not be doing it at midday, because both of these are the times when they're most likely to be asleep. if I'm trying to test its climbing ability, for example, I should try to use that time window when I know flies are quite active. Most importantly, I should do my experimentals and the controls at the same time.
I see. Yeah, I guess irrespective of what time is chosen, it should be same for the control and experiment, and that's how you deal with it, right?
Yeah. And ideally, keep them in a nice light dark cycle.
Yeah.
Keep all your cultures in a nice light dark cycle if you want to do behavioral experiments.
So I have a follow-up question on this, because you just said behavioral experiments, but I'm just wondering whether even to sacrifice the animal for, let's say, Western blot or something,
definitely. So
if many of the things are circadian regulated, and if you're looking at levels, it might make sense, right, to also do it at the same time.
Yeah, definitely. Everything must be done at the same time. Like I said, definitely, that is for sure. Now, I think the question is some people have is, should I keep them under a strict light dark cycle or not? Because what happens if you have kept them on continuous light, fly rhythms break down in continuous light. I'm sorry, I did not mention it while I was speaking. In continuous darkness, fly rhythms will continue to happen. But many fly rhythms break down under continuous light. So, if by breaking down the continuous, breaking down the core clock, have you actually destroyed the rhythms of many other physiologies? I don't know. Why I'm saying that is, for example, the egg laying rhythm will continue to happen even under continuous light. But I know that because I determined that to begin with. So if I haven't found out, then I better make sure that the phenomenon I am doing, that I am interested in, is actually flat across time of day. And then it's okay. I can keep the whole incubator or the room in continuous light. On the other hand, if under a normal situation it is cyclic, but simply by keeping it on, you have broken the whole rhythm down, and that has an impact on the physiology, then your controls and your experimentals are showing you something which is not true, which is not relevant maybe for the fly.
So just to clarify, what does it mean by breaking off cycle? That means all the flies in that vial will have different cycle, or there is no cycle?
In terms of the clocks that we understand in the fly, the rhythm is broken in every fly. Because what happens when the light is on, cryptochrome is on, TIM is degraded, so there is no cycling of per and TIM. So the clock that we understand of that kind is broken. But like I'm saying, I can say for egg laying rhythm, it doesn't matter. You can have the lights on, but you will still see a nice rhythm in egg laying. We don't know about other things. Activity will break down for individual flies.
I have a question.
Go ahead.
So, since you're saying that there are clocks within the peripheral system, as in peripheral clocks, how nutrition actually plays a role in, say, for example, a high lipid fed fly, how does it have its rhythmicity behavior as compared to say sucrose fed fly?
Okay, so from what I understood, you're saying high fat versus high sucrose, but both are supplied ad libitum, right?
Yeah.
Okay. Clocks are supposed to be nutrition compensated, just as they are temperature compensated. Okay, that is one more definition for a clock. But what I think would be more interesting is if you have cycles of food availability. That we know can affect fat body clocks. Okay, so what I'm saying is not high fat all the time, but high fat during the day alone, or high fat during the night alone. So if you provide cyclicity in nutrition, it strongly impacts the cyclicity in the fat bodies. Now, how strongly does the clock in the fat bodies impact the central clock? It turns out not so much. So somehow the central clocks seem to be kind of immune to such information from the peripheral clocks.
Okay. Okay, thank you.
So Anurag is asking whether exposure to artificial light also is cyclic. For example, if you were to, I mean, the screen light exposure, like mobile phones and laptop screen, also affect human sleep wake cycles.
Yeah, so for a long time it was, there were arguments about this because there is a huge blue wavelength component in lights that are emitted by many devices, and a study by a very well respected group in Harvard led by Chuck Seisler showed that indeed the blue light that is emanated from these kind of devices can reset the clock of humans such that they have higher latencies to be able to fall asleep. Because of that study, which came out about 4 years ago, if I'm not mistaken, or maybe, yeah, about 4 years ago, maybe around that time, many of these device manufacturers have what they call a nighttime mode, which they claim is where they set off filters, which make sure that after evening, those kind of wavelengths are cut off from the screens. Yeah, so it's not just about light per se, but also the spectrum, and the dawn and the dusk spectrum of real world light is actually potent at resetting our clocks. Vertebrate clocks, human clocks, mammalian clocks are particularly sensitive to certain wavelengths, especially at dawn and at dusk.
Thank you, ma'am.
Are there any more questions? Okay, if there are no more questions, let's thank Sheeba for
Thank you guys.
Wonderful talk. I will be listening the first part of the talk because we have recorded it. I couldn't attend it. Yeah.
Sorry for the many
thank you very much. I mean, yeah, I mean, these are unavoidable. You couldn't do anything about it. So it's all good. Thank you very much. And we will, it was very interesting, and there were a lot of, I mean, this is applied, right? So it was very, very good, Sheeba. Thank you very much.
Yeah, I was going to say that anybody needs to ask questions, you're welcome to email me as well. Yeah, even later on. Yeah, thank you. Bye-bye. Thank you for your enthusiasm.
Thank you.
Bye-bye.
Bye.